My name is Mike Tisza. I am a postdoc at the NIAID in Bethesda, Maryland. I wrote Cenote-Taker2, this tool, during my PhD, but I continue to maintain and update it. So I hope it can be useful. I'm trying to approach this presentation as kind of from the lens of, what do people at the workshop want? And so hopefully I can provide some useful details here. In 15 minutes, I can't be too extensive, but some useful details, some opinions about how one might want to conduct a virome survey. At the end of the day, Cenote-Taker2 can be used for predicting and or annotating virus sequences. I think the thing I'm going to say first and the thing I'm going to say last is that all virus and prediction and identification tools are a little bit different. They're going to give you a little bit of a different output, and they're all a little bit wrong. And so I hope that part of the point of this workshop is that we can exploit the ways in which they're a little bit different to give us more interesting data, while we can also minimize the way in which they're a little bit wrong by carefully inspecting the results. Pros and cons of Cenote-Taker2. It does discover all types of viruses. So that's phages, are eukaryotic viruses, double-stranded DNA, single-stranded DNA, and RNA viruses. It can take many input data types, including prokaryotic genomes, metagenomic assemblies, virome assemblies, of course, and meta or normal transcriptomes, if you want to look for RNA viruses. It does, in my opinion, make nice maps and annotations of various features on these virus sequences, and allows you, importantly, to inspect the outputs of Cenote-Taker2. You can also feed the resulting contigs from another tool into Cenote-Taker2 just to make the annotations if you want. The cons are it's a lot to install. It's like 130 gigabytes of files that you need to install. You probably need an HPC to do this. But you can use the submodule called Unlimited Breadsticks on GitHub. You can find that on GitHub, and that's much smaller, and it's just more based on the discovery part of it. And there's a pro/con, I'm not really sure which, is that it doesn't use machine learning. And I think it's a con because I think a lot of smart people have figured out machine learning, and they're using it to find out really interesting things. And I think it's a pro because I don't know how to use machine learning, and I'd probably screw something up. So I use a different strategy, which I'll talk a little bit about in a couple slides. But I think here's the kind of the guiding questions that I think people have about virus discovery and virus detection projects. And I hope that Cenote-Taker2 can fulfill some of these questions. So I think people ask, how can I identify viruses in my data or the data that I'm going to generate? How can I extract integrated virus genomes from the flanking bacterial chromosomes? Karthik talked a lot about prophages in his talk. How can I draw nice genome maps for my virus genomes? Those can be useful just for a figure in a paper. You can deposit the outputs from Cenote-Taker2 in GenBank, but also they're very important for inspecting for imputative or possible false positives in your data. And then, of course, people want to know how you can validate hits or predictions from different virus identification tools. And I think I'll talk a little bit about that. The guiding principle behind Cenote-Taker2 comes from kind of just sequence gazing at virus genomes. So I'm going to give an example of what a hypothetical large double-stranded DNA virus genome looks like, broken down into its components, just based on the functionality of the genes. So in this hypothetical genome map, you would have genes function probably for integration, right? An integrase, replication like a DNA polymerase or certain helicases. You have, of course, because it's a virus, it has virion proteins like major capsid proteins and packaging proteins like terminases. There's usually genes to control the lysis-lysogeny decision like lysins. And then most of these genomes also have these kind of sandbox areas, which tend to consist of a lot of hypothetical proteins, but could also be things like DNA methylases and anti-CRISPRs and auxiliary metabolic genes, for example. And I think these different kinds of genes within virus genomes provide very different levels of usefulness for virus discovery. So first of all, I think because viruses must encode a virion gene, and usually they encode a packaging gene, and these are also basically unique to viruses. The virion genes and the packaging genes are very useful for discovering new related viruses. And so these get the happy face. And virus genomes basically always encode replication proteins, and they tend to be pretty unique from other mobile genetic elements, but certain plasmid and ice type elements can have somewhat similar replication genes. So those can't always be exactly discriminatory, but they can be useful. All mobile genetic elements have integrases, so that's not useful. Lysins and holins, for example, in the lysis lysogeny section, those have cellular homologs oftentimes, so I don't find them that useful. And then the sandbox region, although sometimes you have a hypothetical protein that's very specific to viruses, I don't find that to be quite useful, very useful that often. Okay, and so I'll talk a little bit about how Cenote-Taker2 uses virus hallmark gene models, virus hallmark gene model databases to search for new viruses in your data. So it begins with, you would take some kind of protein family, let's say like a major capsid protein, you make a multiple sequence alignment of its amino acids. You can do structural prediction there, and you can find which domain within the larger multiple sequence alignment actually corresponds to that major capsid fold. And then you can take that domain, get rid of the other domains that may or may not be there, and generate a hidden Markov model there. It's important to remove those other domains because they can give you some false positives because they might be an unrelated protein domain. And then, basically from there, you'd have your contigs or your genome, you extract and translate the ORFs, and then you query those hidden Markov models, which are just these statistical models of gene families, basically with Hammer or Hummer. And basically that's where you get your basal set of viruses. Now, because this is phage-based, I want to talk about prophage pruning a little bit and how that works with Cenote-Taker2. This large figure here actually is a plot that was generated from a contig that I fed into Cenote-Taker2. But basically what you have is you have a larger contig, and then each of the ORFs on that contig gets categorizes in one of 4 categories, either virus hallmark gene, common virus genes that are not hallmarks, unknown or hypothetical proteins, and then all other genes. All other genes are mostly bacterial, and so those get negative scores, but these other types of genes, they get positive scores. And you basically, Cenote-Taker2 generates a sliding window score, and you get these large chunks of multiple windows that have a positive score, but they also require a virus hallmark gene. So this area here would become a putative prophage, but this little one over here, that's not a prophage because it doesn't have any viral hallmark genes. As far as the accuracy of prophage prediction and trimming those ends exactly, I think this is not an exact science. This is probably the hardest thing to do, and Karthik talked a little bit about it, but I do want to compare a few different tools on some data that I think is compelling. So I think it's interesting to compare with actual experimental data if we can get it. So what I did was I went into the SRA to look for public data where some experimentalists somewhere did a prophage induction on a bacterial reference strain, usually by a mitomycin C or something, and then they collected their phage particles, and then they sequenced the phage particles. They deposited those sequences, which you can map back to the bacterial reference genome to find exactly where that phage is in the genome. And so comparing, and so here's sort of an IGV view here where we have just because this mountainous region here is where the prophage is, and this is where it ends and begins from a Bacteroides vulgatus genome. You have different prophage prediction tools, Cenote-Taker2, which is my tool, Virosorter2, Vibrant, and CheckV. What you can see is none of them are exactly right. Cenote-Taker2 in this particular example does quite well. Virosorter2 has a bit of a hang off on the right. Vibrant is a little bit of a undercut on the right, and CheckV, which is a really great tool, has a little bit of a undercut on the left. And I'll look at one more example just because I think I don't want to take up too much time. And this is a same type of experiment that someone did with Salmonella enterica. And this is the prophage here. So Cenote-Taker2, as you can see, does an okay job. It has some overhangs both on the left and the right side. So, this is not calling the exact border. Virosorter2 is a little bit off. Vibrant and CheckV in this case are actually like almost exactly correct. So those in this case are a little bit better than Cenote-Taker2. This is, it's just 2 little examples, and really to get any sort of meaningful qualitative or quantitative measurement, we would have to look at a lot of these. But I do think it's important to kind of look at these, and you can see that's, you know, if you look at different genomes, different tools will perform slightly differently. And so when you're doing your project, it can be important to compare the different kind of calls that you get from these different tools. Okay, I think I have a little time. So yeah, I do want to talk about virus genome annotation. I'll quickly go through how it works with Cenote-Taker2, and I think I'll show a couple of examples of how that can be useful for someone who wants to use it. So after the virus prediction occurs, you feed that contig. That contig gets fed into the annotation part of the pipeline. So if you want to, you can use BLASTN to find if that contig is, you know, very closely related to some phage or bacterial chromosome in GenBank, so that you know that, you know, maybe you're finding a new strain of a previously identified phage or, excuse me, or virus. It identifies tRNAs, and then it does a threefold iterative annotation for all the open reading frames. So first, uses Hammer to query the custom viral HMMs that I made. It does RPS BLAST to query CDD database, which is conserved domain database. And it also has an optional module to use HH Suite, which is like HHpred, against a variety of databases, which provides really good functional annotations for very divergent proteins. And after that, after all of the annotations are in, Cenote-Taker2 will do taxonomy on the contig, and this is probably not as good or as in-depth as some of the things we hear about later in the conference, but basically it uses a strategy to find the best open reading frame. So if this was a double-stranded DNA phage, it would find the portal protein or the terminase protein or the major capsid protein to use to query a RefSeq to find what the closest hit taxonomically is, or if it's an RNA virus, it would use the RNA-dependent RNA polymerase. So that's what it means by best ORF. And then, you know, that gets all wrapped up together into a genome map, as well as some other files that you can, again, submit to GenBank and do other things with. So I do want to talk about inspecting the genome maps, right? I think this is like a really useful technique, and it can really allow you to figure out if the putative virus that you have is really a virus or not, because sometimes, even though these tools are very sophisticated, it's better just to use your own eyes. And so I did a quick annotation of a metagenome with one of the other tools that I talked about, and then just annotated it with Cenote-Taker2. And so this is a really nice virus genome. There are some very telling features about why it's nice. First of all, it's a circular genome of about 40 kb. It has this whole area is just filled with these virion proteins, portal protein terminase, major capsid, all these tail proteins. And so if you see things like this, I think that's a really good indication that what you have is a virus, and you don't have to worry about it. You can, of course, take all of these open reading frames and query them in some other way with HHpred or something, just to make sure. But then, you know, just like every tool has, I did find a false positive when I was looking through this data set. And I won't say what tool it is because it doesn't really matter. But, you know, so this contig here, it's about 28 kb. It's got some replication proteins, but it didn't have any virion proteins, and it had some conjugation / type 4 excretion system proteins. And so if I saw that, I would probably discard it from my data set for my future analysis. And I just bring this up as just like a single example, just as how you might kind of best paw through your data a little bit by using genome maps. Okay, and so that's pretty much what I was going to talk about. I think I got it in under time. So I'll say it again, all virus prediction and identification tools are a little bit different, and they're all a little bit wrong. And so it's important to exploit all their differences and minimize all the things that they get wrong. And that includes Cenote-Taker2. And so I hope that helps with whatever projects people have going on. Cenote-Taker2 uses virus hallmark gene models to find virus sequences in many data types, can prune prophage as well. It makes nice genome maps that you can inspect, and it also facilitates submission to GenBank. Please, though, I would hope that everyone reads the docs on GitHub for the recommended settings for your data type so that you get the best results. And I do want to acknowledge all of my co-authors, and especially I would acknowledge everyone here for listening in. And I will, of course, course, happy to take any questions. So thank you.